Yeah, thank you everyone for joining us today. Today is the 30th November 2021, and this is actually our last WebEV talk for this year. Looking forward to a little bit of break in December, and likely to start on the end of January or early February. So today we have Kirsi Rilla, who's an associate professor of cell biology, Institute of Biomedicine at University of Eastern Finland. So thank you, Kirsi, for being our last speaker with such interesting topic today. So Kirsi has a long history on the biology of the extracellular matrix, especially hyaluronan, and her group's pioneering findings on hyaluronan-induced philopodia. Hyaluronan-coated EVs are well recognized. The current focus of her team is to understand the biogenesis of EVs and their interaction with the extracellular matrix and target cells, with special interest to utilize the unique properties of hyaluronan and its specific interaction with CD44 to engineer and exploit EVs as future vehicles of therapy. So Rilla Group utilizes 3D cultures as research models and modern bioimaging techniques and their combination for EV detections and analysis. So today, her talk will be about diverse plasma membrane protrusion act as platforms for extracellular vesicle shedding. So before we start, I'd like to thank Horiba Scientific, who's been supporting us since May, and they have been generously sponsor this activity until today. So that's really great. Thank you so much. And starting from next year, we're going to start looking for different sponsorship to continue the WebEV talk. So if there's any other company or industry or non-profit organization who want to sponsor WebEV talk, please contact me at webevtalk@gmail.com. So with that, I'll stop my screen share.
Thank you, Carolina. So I will talk about these diverse plasma membrane protrusions as a platforms for extracellular vesicle shedding. I will start with some kind of introduction to imaging methods in EV research. Then I will continue to tell a story about hyaluronan-coated philopodia and EVs. And then I will talk about these plasma membrane protrusions at platforms for EV shedding, and finally, some examples how we are going to utilize hyaluronan to enhance the potential of EVs in the future as a therapeutic tools. So we will start here. So we believe in the power of imaging in EV research. And here are some examples. For example, here we have grown these breast cancer cells inside a three-dimensional matrix, and we can clearly see how these cells produce the vesicles outside in the matrix. And here are some examples of the most common imaging methods. With light electron microscopy, we can image with fluorescent markers, the EVs in populations, also single EVs if we use a high or super resolution, and also shedding and uptake. But of course, with the light microscopy, the resolution is not so good as in electron microscopy. And if we want to go to higher details, we utilize microscopy or scanning electron microscopy to see the surface structures, for example. And the best thing comes out when we combine these different techniques. So CLEM means correlative light and electron microscopy, where we kind of combine light and electron microscopy, and we can image the same vesicle, for example, first with the electron microscopy and then with light microscopy, and we can combine these images and see much more than we could see with either of these methods. And immunoelectron microscopy is also good if you really want to see in high resolution and also label specific proteins. And this is an example of live cell imaging. Here is the plasma membrane of a breast cancer cell in just a small portion of the plasma membrane. And we will see here in a time-lapse movie that vesicles are shedding from the plasma membranes in different areas here. And from this series of still images, it's even more easy to see, for example, here, how the tip of this protrusion is shedding a vesicle here. And here is in a little bit more detail, this correlative electron and light microscopy. So we use this kind of gridded cover slips where we have these numbers and letters. And by this way, we are able to find the same cell again. So we first image the area in a light microscopy, for example, with confocal microscopy as here, and then we process the sample for electron microscopy, and by using this grid we find the same cell and we can combine the images and we can see much details here, for example this filopodia here or this kind of vesicle-like structure. But there are some challenges on this imaging of EVs or filopodia that I'm going to talk today also. And I will now introduce some of these challenges. And first is this fixation. So here is the same cell before fixation and after fixation. And we can see a clear difference. For example, these filopodia that are very clearly seen on this live cell and also in the side view are mostly disappeared after fixation. So if we really want to see the original image or how the cells really behave, the fixation is not a good thing. One challenge in this live cell imaging is phototoxicity. And especially when you want to study vesicles, it's difficult because, for example, here we have imaged this couple of cells for 20 minutes. And there really seems to be some phototoxicity because you see that filopodia are swelling and they are kind of forming very, very big vesicles that are shedding off. And here you can see the same as still images. So these cells are really going to die here, not shedding. Okay, they are shedding vesicles, but at the same time, they will die. One more challenge is this two-dimensional versus three-dimensional imaging. Here we see a series of optical sections from a single breast cancer cell. And for example, in this section, you see just spots. And from this, you are not able to say if these are maybe vesicles or some other structures. But when we do these 3D projections, we see that actually these spots that we see here are cross sections from this filopodia. And one more example on this 2D. Here we have cultured these cells in 3D cultures. But this first example is an optical section. So similar as before, just from one layer of the cell. And again, from these spots that we see here, we are not able to say for sure if these are just cross sections from some cellular parts or are they really vesicles that have been shedding away from the cell. But three-dimensional projection reveals that actually in this case we have both. We have a filopodia protruding from the plasma membrane of this cell, and then we have also vesicles that are really separate from the cell. And then one thing about these uptake studies, of course, there are also very many problems with labels, but here we have used GFP has 3 as a label. So the donor cells express this plasma membrane protein, and that's why all the vesicles that we isolate kind of carry this GFP has 3 label on their plasma membrane. And when we treat target cells with these EVs, we see that there is plenty of EVs that have been bound to target cell. And it seems also here that they are in the cytoplasm and even in the nucleus. In this another image, we have stained CD44 to visualize the CD44 protein, and also at the same time, we can see the plasma membrane. But when we do a confocal analysis and the kind of jet section, we kind of cut the cell, we actually see that there is nothing in the nucleus, but the vesicles that we see are on the top of the nucleus, and most of them are actually on the plasma membrane. Only some signal can be seen in the cytoplasm. And then the same thing in electron microscopic level. This is a 3D culture of breast cancer cells, so actually similar cultures that we saw in these confocal images. And here we can see similarly filopodia clearly, and then we have also these vesicle-like structures, same problem again. And in electron microscopy, we have even thinner kind of sections, so it's very difficult to really see what we have there. And a similar case in this another picture. And for this, the only way to solve this is also to 3D EM. So here we have done a tomography, and from the series of sections made a kind of 3-dimensional illustration. So here we can see that in this case, we have a group of vesicles of very different sizes. And we have been able to also count or analyze the size distribution from these images. So actually we see much higher amount of smaller vesicles than usually seen, for example, in NTA analysis. And another thing that we can do is scanning electron microscopy, and this is from the same cultures as before, but we see a little bit different things because we see these kind of surface structures only. There are many restrictions on scanning electron microscopy as compared to this transmission electron microscopy, but in some cases, this is also a good way to visualize vesicles or cells that produce them. Okay, that was a short introduction to these imaging methods. And then I will tell this story about hyaluronan-coated filopodia and EVs. You see that it's a very long story. It started already when I was doing my PhD, but it still continues. So hyaluronan, or HA, or hyaluronic acid, is a natural glycosaminoglycan of our body. And one of the richest sources of hyaluronan is rooster comb, where it was traditionally kind of isolated, but nowadays it can be also produced in bacteria. But hyaluronan has very many different applications in biomedicine. It can be used as a painkiller in osteoarthritis. It's widely used in cosmetics as a kind of filler, but also topical ways to use it. And for example, in moisturizing eye drops. And another very rich source of hyaluronan is vitreous body of our eye. So here you can see that hyaluronan is very transparent. And it's also biocompatible, non-toxic, non-immunogenic, and acts as an ideal biological lubricant. So it has many different applications. And hyaluronan is produced in a very unique way. This enzyme that produces hyaluronan is located on the plasma membrane. And it utilizes these two sugars, glucuronic acid and an acetylglucosamine. And at the same time, when it synthesizes this chain of sugars, it protrudes it to the extracellular space. And CD44 is the most important and most studied receptor on the plasma membrane that binds specifically and with high affinity hyaluronan. And typically the cells that produce high amounts of hyaluronan have this kind of hyaluronan coating around them. So it's a kind of glycocalyx on the plasma membrane. And here we have used a specific probe, a fluorescent hyaluronan binding complex that very specifically detects hyaluronan, and we can use it also in live cells as we have done here. But then when we studied this hyaluronan synthase, this enzyme mechanisms, we found that when we overexpress this hyaluronan synthase enzyme, the cells start to grow this kind of very long apical filopodia. In the beginning, we didn't know how to call them. We maybe called in the first publications, they were microvilli, but nowadays we know that these protrusions resemble filopodia, and they also exist in vivo. And they act as a kind of hyaluronan factories. So there is a high hyaluronan synthase activity on the plasma membranes of these protrusions. And here we can see a live cell that is overexpressing the GFP labeled Has3 enzyme. So we see this filopodia, and we see this hyaluronan coat labeled as red in the beginning, but during this video, we have added hyaluronidase enzyme in this cell culture, which digests hyaluronan away. So we see that the red color disappears. And that results in the disappearance of this filopodia also. And we believe that these filopodia are dependent also on hyaluronan synthesis and hyaluronan coating around them. So here we can see from still images how the same cell looks before this enzymatic treatment and after. So the filopodia disappear or collapse. So as a summary, we can say that when we remove hyaluronan or inactivate this Has enzyme, we have done many different experiments with different treatments, but in all cases, these filopodia collapse or disappear. So hyaluronan acts as a kind of extracellular cytoskeleton for this filopodia. And here is one example. Here we have used different glucose concentrations in the culture media of these breast cancer cells. And in the glucose deprivation, the filopodia do not grow because the cells are not able to produce hyaluronan anymore. But when we increase the concentration, the filopodia and hyaluronan secretion are kind of normal. But here, actually in this upper, in this A, we have live cells, but here in E, we have fixed cells, and we see here that the kind of effect disappears here because the filopodia are not resistant. They are not. They do not like fixation. They disappear. Okay, but then how we came into EV field. So This is one example from the time when we were studying these GFP has 3 overexpressing cells. And here we have a chondrosarcoma cell in the middle. And we were wondering, what are these spots around these cells? We didn't know anything about EVs then. And when we added this hyaluronan probe, we saw that actually all these spots also have a hyaluronan coating around them. And then we started to look at the literature and found that they may be microvesicles. And we started to study what happens and found out that when we overexpress this GFP has 3, also EV secretion is increased. So this hyaluronan synthesis accelerates EV shedding. And another thing that we found was that these EVs are shedding from tips of this hyaluronan-coated phylopodia. And later we have found that it's not only this overexpression of hyaluronan synthase, also cells that are producing hyaluronan naturally in high amounts, like stem cells, produce these kind of HA phylopodia and HA EVs. And they can also mediate melanoma signaling. So, as a summary here, we can say that hyaluronan synthesis induces EV shedding. And many of these EVs are shedding from the tips of phylopodia or from retraction fibers, for example. Okay, then a little bit more about these plasma membrane protrusions, phylopodia, and so on, that act as platforms for EV shedding. And this publication maybe was the main reason why I was asked to have this presentation today. It was published a couple of months ago in JEV. So it tells about the diverse plasma membrane protrusions that act as platforms for extracellular vesicles. But let's see in more detail. So when looking for previous publications, even these very early findings suggest that actually many of these vesicles are originating from different protrusions. This image is from this Wolf publication with platelet dust. And this is an activated platelet. We see that they have these very long protrusions with these kind of tip structures. And it seems that the same mechanism actually is acting here. And this another example is from this white paper with erythrocytes, and a similar phenomenon can be seen here. And later also similar findings are from chondrocytes, for example, and osteoblasts. And here we talk about matrix vesicles, which is one sub group of EVs. And also microvilli of enterocytes have these tips that generate EVs. And here are more examples. So when you go through the literature, there are many, many examples on different protrusions that produce EVs. The nomenclature for these protrusions is kind of variable as well as in EVs. So sometimes you call them microvilli or phylopodia, as we did in the beginning, or cytoplasmic processes. But in many cases, they are similar actin-based protrusions. So there are many examples, but nobody has really kind of concentrated of this aim. And that's why I thought it was time to kind of write this review article. And then I will show some examples of these different protrusions in our own studies. Here is a simple budding from plasma membrane. Here we can see a kind of different sizes of EVs in the same figure. In this one, we see kind of purling of a protrusion or a retraction fiber as here. Okay, a similar example here also. This kind of bigger protrusions that have a kind of small, many small EVs can be called microsomes. And in the bottom, we have these very, very long protrusions that are usually called tunneling nanotubes. And very often when we see a kind of tip of this kind of long nanotube, we can see vesicles there. So they can kind of go to another cell and attach there. And this is one example of these tunneling nanotubes with the confocal microscopy. Here we have a similar GFP-Has3 overexpressing cell with hyaluronan coating. And we see that these protrusions from this cell go to this cell, which is negative in this case. And it has kind of left these vesicles on the plasma membrane of another cell. So this may be one mechanism how the cells change information, kind of. It can be called a juxtacrine signaling here. And here is the same in scanning electron microscopy. This is an example from a live human mesenchymal stem cell. And here we have similarly labeled this hyaluronan coat. And if you see the area here, you see that this protrusion is kind of retracting here. And it leaves these vesicles behind. So we can call them retraction fibers or kind of footprints that cells leave on the bottom of the plate when they move. And here we can see in more detail these vesicles. So most of them have this hyaluronan coat. Some of them have this kind of spotted structure, which suggests that these are really originating from these protrusions. And one more example. Here we have labeled CD44, which was this hyaluronan receptor. We see that these vesicles that form this kind of purling mechanism have also CD44. And then when cells migrate or move, they kind of leave these trails behind. And in this manuscript, which is not published yet, we have used MCF10 CA cells. And these are not overexpressing this Has enzyme. They naturally produce high amounts of hyaluronan. And here we have used the CD9 antibody to label these cells, and we see that CD44 is accumulated on the plasma membrane and also in these filopodia-like protrusions. And we see that the EVs that are left behind in these trails contain CD9. And these trails also have a hyaluronan coating. So we believe that these cells utilize these kind of EV-rich, HA-rich trails for collective migration. So, as a summary, we can say that EV originate from diverse protrusions, but how those EVs then differ from other EVs that are not coming from protrusions, such as exosomes. We have analyzed these EVs from a GFP-Has overexpressing and non-overexpressing cells. And here we can see that, of course, okay, the negative cells do not have this GFP. Okay, the negative EVs do not have this GFP-Has3. but they carry all the normal markers that we have for EVs. They have CD44, CD9, CD63, and actin. And here we have compared the size distribution. It's similar, but there is a more increase in the smaller EVs. And I haven't mentioned before that, but this filopodia diameter is around 100 or 130 nanometers. So the vesicles that are shedding from filopodia are most probably in this group here, between 100 and 200 nanometers. And in transmission electron microscopy, we really don't see any specific differences. They look almost the same. Maybe this GFP has 3 induced have more kind of this stuff around, which may be hyaluronan. And in these markers, CD63 and CD9, we haven't seen any special differences, at least yet. And this is from a scanning electron microscopy of the same vesicles. We see these big vesicles also sometimes, but the surface structure is kind of similar, and the overall view is similar. Of course, we see these kind of aggregates that originate from ultracentrifugation processes. So it will be very challenging to really analyze these vesicles that are shedding from protrusions because there are no specific markers yet how to kind of separate them from exosomes or other vesicles. But this is a kind of summary how these different protrusion-derived EVs are shedding or pearling, or sometimes or scission may happen. Okay, then we come to the last part of my presentation. So some examples of our recent data and how in the future we are going to try to utilize hyaluronan to enhance the potential of EVs as therapeutic tools. So it seems that hyaluronan is involved in every stage. So it accelerates the biogenesis of EVs. And when a single vesicle have this hyaluronan coat around it, it kind of protects the vesicles from degradation, and it increases the circulation time, for example. And hyaluronan can regulate also the biodistribution of EVs. We don't know much yet, but in the future we are going to study that in more detail. And because of these hyaluronan CD44 interactions, it also regulates the uptake or binding of EVs to the target cell. So hyaluronan protects EVs and increases the circulation time via immunoprotection, via resisting osmotic changes, for example, and also regulates the binding. And here in this very high resolution image, you can see that, here we have labeled plasma membrane with cell mask. So we see the single vesicles here, and here we have hyaluronan. We can see that the coat of hyaluronan may be sometimes very huge here. So actually the size of a single vesicle can be 10 times bigger with hyaluronan coat than without it. And now we have learned how to engineer the donor cells. So we can express this hyaluronan synthase to increase hyaluronan synthesis, or we can also express CD44 in the donor cells. So it kind of binds hyaluronan there, because we know that the CD44 is also carried by vesicles. And this graph here shows that also because hyaluronan has a negative charge, it increases the zeta potential of EV, so it increases the negative charge, which may affect the EV use in therapeutics, for example. And here is an example how we have studied the CD44 HA interactions in EV binding. So we have used this correlative light and electron microscopy here. And we have used the same. GFP has 3 vesicles that can be seen on the plasma membrane of this target cell. And we have stained CD44 here, and we see that these vesicles like to bind to areas where CD44 expression is high. And scanning electron microscopy reveals that we really have vesicles here. This vesicle comes from here, and this single vesicle is from here. And this intensity profile shows that the GFP has 3 from vesicles and CD44 from cells are kind of colocalizing. Of course, in this case, CD44 can also be originating from the vesicle itself. And then we have treated these cells with hyaluronan oligosaccharides. These are kind of short hyaluronan chains that bind to CD44. So they kind of block CD44 binding. And we can see that at least partly the EV binding is inhibited. And then we have continued these 3D cultures. We know that the isolation of EVs from kind of animal-derived collagen-rich gels is challenging, and there are also contaminants from animal-derived matrices. So we have started to use this nanofibrillar cellulose that is extracted from Finnish wood. Actually, you can see on my background that in Finland we have lots of wood, so that's very easy for us. So this is totally plant-based matrix, and we grow these tumor cell spheroids. Now we have done 7 days cultures, and we can image the cells in live cell imaging because this cellulose has a very good kind of imaging properties. It doesn't have any autofluorescence, for example. And after that, we can enzymatically digest the cellulose away. And after that, we can isolate EVs as normally, for example, with differential centrifugation or any other method. And then they are ready for downstream analysis. We have analyzed here the particle concentrations and size distributions with NTA analysis and with imaging with confocal and electron microscopy. And we have utilized this model to study, for example, this hyaluronan things. So here is one more example of this nanocellulose cell culture with lots of vesicles secreted by this cluster of cells around them. And here are the same vesicles after the isolation. And we see the same effect in these 3D cultures. So when we express this HAS3, the concentration of particles is increased. But interesting is that when we add extra hyaluronan to this matrix, actually the particles are decreased. So it seems that the vesicle secretion is decreased. So we don't know yet how this HA affects here, but we are continuing these experiments. And the last example is our future aim. So we are going to study how we could utilize these HA coated EVs for targeted drug delivery. And as a control, we use hyaluronan coated liposomes that are already widely used as a research. And then we compare them with these hyaluronan coated EVs. And we use, for example, CD44 positive and CD44 negative cells as targets to study in more detail these interactions between EVs and CD44 receptors. And we really believe that hyaluronan has a dual role, so it can protect EVs, but also act as a specific targeting moiety. Okay, finally, I would like to thank all the collaborators. There are so many during these years that I didn't want to mention the names now, but especially today, I would like to thank all my current and previous group members and also funding. So thanks, and I'm happy to answer your questions or comments.
Okay. All right. Let's just stop there. Yeah. Thank you so much, Kirsi. That's amazing presentations, and also I'm really amazed of how we can really dissect all this plasma membrane protrusion. So I think my curiosity is like, if you really want to dissect them, how would it be best to look into? I mean, the transmission electron microscopy is kind of still images, and live imaging is live imaging. But could you give us more advice on how to start looking into that as a beginner, looking into these images?
Yeah, of course, light microscopy is more kind of easy and less expensive than transmission electron microscopy, and most labs have it. Maybe electron microscopy is not so common. So I would advise to start with light microscopy. And of course, it's quicker and so on. It depends what you want to see. If you really want to see details, of course, electron microscopy is the better choice, but light microscopy is more easy, and you can also label these proteins more easily.
How about this differentiating between those microvilli or philopodia? Do you have a sort of quick way to differentiate them?
Yeah, okay. Microvilli are more stable structures, and they are typically expressed in cells that are kind of they absorb something, like in our gut, in intestine, these enterocytes, or in kidneys, epithelial cells that absorb this urine. So they are usually a little bit shorter and stable, but they have a similar actin-based structure as philopodia. And philopodia are more kind of, they move and they grow and retract very fast. But sometimes it's not so easy to separate them.
Yeah, we have a few questions here. So I'm starting with Tatini. Could you please start asking your questions? Tatini? I think you're already unmute there. Okay, I'll just read it. How long are these HA polymers in terms of molecular weight or chain length in nanometer attached on the EV outer surface?
Okay, in theory, HA polymers can be very different lengths. We talk about short hyaluronan chains and long or high molecular and low molecular weight. And as the longest form hyaluronan can reach even 25 micrometers, if I remember right, but they can be very, very long. And you saw this one example where it was several, let's see, it was maybe 2 micrometers, I think, maybe at least 1 micrometer. So very, very long. So it can be even longer than the EV diameter itself. But to measure these, it's very challenging because we don't yet have a good method to kind of, we can analyze the length of hyaluronan with chromatographic methods, but they are not sensitive enough that we could do the same analysis from EVs. We can analyze it from cells or from the culture media of cell cultures, but not EVs yet. So, but I believe that they can be very long, really, as you saw from these microscopic images.
Yeah. Celeste, your questions, please. Okay. Yeah, so this is like a new settings for the WebEVTalk. So there's an issue in my settings that I apologize for that. But I'll just read the questions now. Have you seen enrichment of cytoskeletal proteins such as actin in the HAS3 plus EV?
Yes, we have seen actin in these EVs. I'm not able to say if it's enrichment. So, but we see that they are active positive. Maybe in microscopy, it's not so easy to detect because this Has protein, for example, it's on the plasma membrane or the membrane of EV, so it's a very kind of high intensity. But actin is in the cytoplasm, and it's not so much. So I believe that sometimes we may have problems to detect all actin positive EVs. But yes, there is actin.
Esther, please.
Yeah, hi, Kirsi. Thank you very much for the nice talk.
Hi, Esther.
I have a question about the formation of the Filopodia-derived extracellular vesicles, because, well, the mechanisms are not fully known yet, but I am actually wondering, is it mainly the pulling force of the Filopodia that makes kind of the vesicle being ruptured from the Filopodia? Or if there was no pulling force, would you then get a similar budding of those vesicles? And I was wondering, does it really matter what kind of matrix the cells grow on? So if they are more stuck to the matrix, do you get a different type of vesicle formation compared to when they are just loosely attached? Do you know anything about this?
Difficult but important question. So we believe that this pulling force is the one mechanism how they form. It can be seen from these videos when the cells are attached to the bottom of the plate and they move a little bit, and then when the cell kind of pulls the Filopodia, some of the vesicles stay there because they may have some, for example, integrin interactions with the substrate, so they stay there. But there is also evidence that some external forces like a kind of shearing stress can induce the shedding. There may be also some active processes dealing with cytoskeleton or plasma membrane changes, but we don't know the exact mechanisms yet. And what was the second part of your question?
Yeah, so
did I answer everything?
If you change the matrix on which they grow,
yeah,
and they're more or less adhesive, do you see differences?
Yeah, I believe that there is differences. The results that I showed in the last slides were just preliminary data. But for example, when we change the density or the matrix stiffness around the cells, there is different amounts of EV secreted. So we can also kind of tune the stiffness of the matrix by putting different concentrations of this nanocellulose or hyaluronan or other molecules. So it will be interesting in the future to study in more detail of these extracellular matrix cell EV philopodia interactions.
Yeah, that's a great system to use indeed, if you can change this density. Yeah, thanks.
Yeah.
Yeah, I think there's a lot of things to learn from this. Sorry, my internet is unstable. Okay, so I have a couple more questions actually. With the hyaluronan, what is the actual functions in the cells, and also why they are on EVs still? Do you observe them always on EVs, or is it just some population has hyaluronan coated, but some not? What do you see?
Okay, hyaluron is a very abundant molecule in our body. So there is more or less hyaluronan almost everywhere, but it's a kind of molecule that supports cell growth. So the tissues that are active in proliferating or healing or cancer are examples where hyaluronan amounts are high. And I believe that if the cell produces hyaluronan, it has always hyaluronan on the plasma membrane. So it can be there bound to this enzyme or bound to these receptors. And if the vesicles is budding from the outer plasma membrane, of course, hyaluronan goes together with the EV. So it may be just accident. So because it's there, it goes to EVs.
Because the abundance.
Yeah, but of course, it can also have this meaning in life. So it can, for example, regulate extracellular matrix composition when we think our tissues in real life. And it can also regulate this targeting, because if there is HA or not, it affects the half-life, circulation time, other properties of the vesicles.
Yeah. Would it be possible, like protecting the vesicles as well, so it's become a stability for the EVs?
Yeah, that's true.
And I think there's one more just technical questions. So I'm really interested to look into those 3D method, 3D culture that you're using, and that was really wonderful image with the vesicles surrounding the culture organoids. But I was just wondering, how can we differentiate using that imaging techniques they're using right now? How can you know that there is not cellular debris, but they are vesicles?
Yeah, it depends what you mean about cellular debris.
Maybe vesicles are cellular debris.
yeah, that I was thinking.
That's a good point.
But with light electron microscopy, of course, you are not able to see such many details. So maybe at least once to confirm with some electron microscopic techniques, you can really see if they are vesicles or something else. But if you have plasma membrane there, then okay, it can be cellular debris also then. So at least actually it was a difficult question.
Well, the vesicles used to debris anyways, isn't it? Come to think about it.
Yeah.
Okay, Georgia, do you want to ask your questions directly to Kirsi, please? Go ahead.
Hi, hello. Congratulations for the presentation. It was really interesting. I don't know if you can hear me.
Yeah, a little bit
Okay.
difficulties, but I will try.
Okay. Since you talk about tissues in general, I was wondering if you, and also cancer, I was also wondering if you analyzed the production of extracellular vesicles from immune cells, or just focused on tissues. Thank you.
My group haven't studied EV production from immune cells, but one of my colleagues, Sanna Pasonen-Seppänen in our university, is studying EV production in monocytes, for example. But I'm not able to say any specific about that.
Okay, thank you so much.
Okay. If you have any more questions, please go ahead now before we close the session. We're also close to the 1 hour mark of our session. No? Okay. Well, with that, I would like to thank Kirsi. Thank you so much for sharing your wonderful presentations today. I really love the images, just amazing, so much things to see and so much fun actually to watch it as well. Thank you so much for sharing that with us. And I think in a very short future there will be a lot of research looking into these plasma membrane derived vesicles and how the biogenesis. So I think what you're doing right now is amazing, and I would like to learn so much from you also in the future with this topic. So thanks so much again, Kirsi. As I mentioned, so this is our last sessions for the WebEVTalk this year. So please have a very good end of your holiday. If you celebrate Christmas, please celebrate it well with your family and friends. And it's a COVID time as well, so please take care. So with that, I hope to see you next year. And if you have any suggestions on what sort of specific topic you want to see at WebEVTalk, just don't hesitate to direct message me, and I'll figure out a way to make the speakers available. So thank you so much. And with that, please take care and goodbye. Thank you, Kirsi.
Thank you. Thank you. Have a nice Christmas. Bye-bye.
Bye-bye.